So hi guys and welcome to the Biomolecules module. So I've laid this one out slightly differently. This module is going to be quite an intense module. We've got a lot to cover. So the way I've laid this out is that we're going to quickly go through the central dogma of biology, some transcription, some translation, and the folding of proteins. But it is something that you should already be aware about. If it's not something you've ever covered before, I really do encourage that you find out more about the central dogma of biology and the folding up of proteins and things like this before you start this module, because this lecture is literally just going to be a very quick recap, ready to prepare us for the amount of practical that we've got to do. So with that being said, we're just going to start off with the central dogma of biology. Now, like I've said, I hope this is something you've come across before, and what you normally see when you see the central dogma of biology is an arrow here, an arrow here, and an arrow here. Now this arrow refers to DNA being self-replicating, and this refers to transcription, and translation. Okay, and obviously the whole central dogma of biology that was coined by Francis Crick is referring to the flow of genetic information within a cell. So DNA is capable of self-replicating, and the flow of information goes from DNA to RNA via transcription, and from RNA into protein via translation. Now, the first thing I thought we'd recap is just the differences between DNA and RNA. Now, the most obvious is simply that DNA is double-stranded, and RNA is single-stranded. Okay, so the next difference is we see the structure of the backbone. Now, the backbone is different for RNA and DNA, but not by much. So we'll just take a quick look at the structure of a backbone of a DNA molecule. Now, this is DNA, and we know because it's double-stranded. And I'll just quickly cover this in the sense that it's something that I hope you've come across before, the 5' and the 3' end. Now, the DNA molecule always runs from 5 to 3, so I would give that an arrow like this, and that an arrow like that. Okay? So DNA always runs from the 5 to 3' end, and it has an anti-parallel other side, which obviously runs also from the 5 to 3' end, but in the other direction, if you see what I mean. So both DNA and RNA have a similar backbone. So they have a phosphodiester bond, which is connected to a sugar, which is then connected to a nucleotide. Now, obviously, with RNA, there is no other side. There is no other anti-parallel strand. It is just a single strand. But DNA obviously does have that other strand. So we have this kind of backbone structure, and the difference is seen within the sugar molecule because both DNA and RNA still have phosphodiester bonds. Now, this one is obviously DNA because it says here it's deoxyribose, but we're just going to have a look at what that actually means. So here we have the deoxyribose and ribose sugar. Now, it can be either because we've got an X here, so we can insert either an OH, which would make it a ribose, or a H, that would make it a deoxyribose, and that is simply the difference between the two. So if it was deoxyribose, I would write a H. So if I gave you this molecule and I said, there's a H here, is this deoxyribose or ribose? You can tell me that this is deoxyribose because it lacks the oxy, which is here. So deoxyribose, DNA, deoxyribose nucleic acid is what DNA stands for, and it has a hydrogen here. Now, if this was an RNA molecule, we have an OH group here, and that is the difference between the DNA and RNA backbone. So this is actually an advantage for DNA because DNA with the H on the second carbon is more stable. The RNA is less stable even. It's more prone to hydrolysis, which can break the molecule. So this is possibly another reason as to why we use DNA as our main storage of genetic information, because it's more stable in a double-stranded molecule, and it is more stable with its backbone structure by having a deoxyribose sugar as part of its backbone, instead of having a ribose sugar that we see in RNA. So then perhaps the next difference we see is the differences in bases. So with DNA, we have A, T, C, and G, and with RNA, we have A, U, C, and G. Now, both of them obviously have 4 nucleotides, but for DNA, so we'll write DNA over here, we'll write RNA over here. So with DNA, adenine pairs with thymine, so you can see here we've got the orange and the red, and I've put an orange and red here, and cytosine binds with guanine, and we've got the purple and blue to display that over there. Now, it does not have uracil. So RNA, it's different. Adenine binds with uracil and does not have thymine, but cytosine and guanine are exactly the same. So that's another difference between the DNA and RNA molecules, is in their base pairs. So we'll just look at that in a little bit more detail. So here are all 5. So this is an A, this is a T, this is a C, this is a G, and this is a U. Now, I've already highlighted the difference between the C and the U, and that's simply that the T has a methyl group over here, and the U does not have a methyl group over here, and that is literally the only difference. Obviously, the 2 nucleotides are split into 2 groups. You've got the pyrimidines and the purines. Now, your purines are A. I'll use a different colour actually, because that might get confusing. A. And G. Your pyrimidines, and I'll use a different colour again, a T, C, and U. So obviously the difference between the pyrimidines and the purines is simply that you have 1 molecule here and you have 2 there. So you've got one ring there, and we have 2 rings here. So you might have already noticed that between A, T, C, and G, or A, U, C, and G, that pyrimidine pairs with the purines. So you have pyrimidine and purine. So if we go with pyrimidine first, so we've got T, that always binds with A, and we've got C, that always binds with G. And obviously also A can bind with U. But with DNA, it's very, very rare for you to get a double-stranded RNA molecule, but it does happen, and I'll show you some examples of that later. So we're moving on from the differences between RNA and DNA now, because obviously DNA, for our purposes, is going to be the only double-stranded molecule. RNA is possible in RNA for nature to be double-stranded, and we sometimes see it in viruses, but for this purpose, we're just going to talk about this in the context of DNA. So between thymine and adenine, we have 2 hydrogen bonds, so this is a hydrogen bond. And between cytosine and guanine, we have 3 hydrogen bonds. Now, the reason this is important is because obviously DNA needs to be a very stable molecule. You can't have it breaking apart for anything, but equally you need it to break apart for DNA replication as well as transcription and translation. So often DNA has a mixture of ATCs and Gs. In any gene, we often find that there's about 50-50 split between the AT, C, and G base pairing. So that makes it the most stable. So you have a nice mixture between the ones that break easier apart and the ones that take more to break apart. So I've just put a final backbone molecule here just so we can go through everything in one go, because I know it's a lot to take in. So we have our phosphodiester bonds here, we have our backbone sugar molecule, and we have our nucleotide bases here. So this could be DNA or RNA, and for this purpose we're going to have it DNA, so this can be a deoxyribose sugar. And we will pair it up, so with C we would have a G, with G we'd have a C, and obviously that would be a triple bond. With A we'd have a T, and with T we'd have an A. So that's kind of the very bare bones of DNA and RNA differences. So we're going to move on to transcription, but I'll do that in a separate video to give you a little bit of a break.